Hey guys, welcome to 18.1 Part 2. We are talking about the Lac Operon. So let me move down here. The Lac Operon is referred to as an inducible operon. This means that when lactose is present, it's going to induce the expression of the gene because the default is set to off. So induce means make work. The cell does not need to break down lactose if it's not present. So it will be stimulated when lactose is present. This is true for most catabolic reactions. When we need to build an enzyme to break down a substance, we only want to build that enzyme when the substance is present. So we don't want to build lactase if there's no lactose present. So therefore, the default is set to off. So lactose is a disaccharide, and it's composed of glucose and galactose, and it is broken down by an enzyme called beta-galactosidase, aka lactase. So normally there are very few of these lactases in an enzyme, or these enzymes, these lactases in the E. coli. So the gene is usually turned off. However, when a person eats food and they have dairy in it, so milk or cheese, that's going to trigger the turning on of the Lac Operon. It's going to induce it to work, and it's going to induce it 1,000-fold within 15 minutes. Because when the E. coli needs to break down dairy, it needs to do it quick through the process of digestion. So there are three enzymes that work for the digestion of lactose. And before those enzymes on the DNA, we have an operator and a promoter region. And so these three genes, the operator and the promoter, make up the Lac Operon. And upstream, outside of the Lac Operon, codes for, once again, exactly the same, an allosteric repressor protein. So a protein that, when is bound to the operator, is going to block the RNA polymerase and switch it off. So our story to this point is the same. The only difference is that the Lac Operon, this repressor protein, is going to stay on the DNA. It's going to continuously repress unless the lactose is present. Whereas in the trip operon, the repressor protein did not stay on. It tended to stay off so that the cell could make tryptophan at all times, unless it didn't need to. So the repressor is going to require a molecule called an inducer. So this is going to induce the expression of the gene. It's going to allosterically inactivate the repressor. So here we have an inactive Lac repressor, and it's going to remove it so that the RNA polymerase can zip along and express the three enzymes that are used for the digestion of lactose. So when the inducer, allolactose, is present, it's going to bind right here. And so allolactose is found in milk. It's found in a very low concentration. It's an isomer of lactose. And that is what is going to bind to the protein. And this should make sense. So when lactose is present, or allolactose is present, it's saying we're going to need to be broken down here. So it's going to trigger the removal of the repressor protein from the operator. So the gene is expressed in the presence of lactose. Okay, I hope that helped, and I would strongly recommend using the list of words that you guys have to draw this out. Let me know if you have any questions.